All right, so welcome everyone. Welcome to the Smithsonian's Hot Topic, Human Origins Today program series. My name is Brianna Pobener, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. Welcome to the third so far in our online Hot Topic program series. For those of you who have registered today, we will be continuing this Hot Topic program series, and you will get an email about our upcoming programs. So this is a monthly series that's hosted with the Human Origins program at the Smithsonian's National Museum of Natural History. One logistical note, this program is being closed captioned. You can navigate to the CC button if you want to turn the closed captions on or off. This will either be at the bottom of your screen or the top of your screen, probably next to the Q&A button. The second logistical note is that if you have questions for our speaker today, who I will introduce in a moment, please type those questions into the Q&A. During the program, which will be about a 30-minute presentation, followed by question and answer, I will be answering any questions that I can in text form in the Q&A behind the scenes. And then for those questions that are still there at the end of Dr. Chirchir's presentation, I will come back on screen and I will be asking those questions to Dr. Chirchir, and she will be answering verbally. So please, if you have any questions about the presentation, about the content, you can put those in the Q&A, which is those two speech bubbles. So again, thank you everybody for attending our Hot Topic program, which is a monthly series that is organized by the Smithsonian's National Museum of Natural History. And I'm very excited to introduce our speaker today. So Dr. Habiba Chirchir is a biological anthropologist and an assistant professor at Marshall University in West Virginia. She's also a research associate with us in the Human Origins program at the National Museum of Natural History. Her research focuses on understanding the relationship between skeletal anatomy and behavior. And specifically, she studies the evolution of modern human skeletal anatomy and how we evolved anatomical features that distinguish us from our ancestors and our similarities in those features with other mammals with whom we share certain behavioral traits. She uses CT scanning technology and X-ray imaging to examine morphological traits of interest in the skeletons of humans and other primates, as well as the skeletons of felids, which are cats, and canids, which are dogs. Habiba received her bachelor's degree from the University of Nairobi in Kenya, her master's degree from New York University, and her PhD from George Washington University. She then completed a postdoctoral fellowship with us in the Human Origins program at the National Museum of Natural History prior to starting her current position at Marshall University. So for me, it's a real special pleasure to welcome Habiba back to the museum today, so to speak, although none of us are actually at the museum. So with that, Habiba will share her screen. She will start her presentation and I will turn my microphone and my video off. So thanks so much, Habiba, for being with us today.
Thank you very much, Brianna, for that introduction. And I am sharing the screen right now. Let me know if it's appearing on your end.
Yep, you're all set. Thank you.
All right. Well, thank you so much for the introduction. It's lovely to be back at the museum, so to speak, with you guys. It's been a while, but this is exciting. So I'm very pleased to be here today to be sharing my work with everyone in attendance. And thank you all of you for joining in to listen to some of my work. Now, as Brianna mentioned, and as you can see from the title of my presentation, I'm going to be talking about bones and how our skeletons have changed over time, especially in relation to our behaviors. In other words, if we are farming, for instance, or if we are hunting and gathering, are our bones stronger or are they lighter? How have they changed as our subsistence strategies have changed? How are they different from those of our ancestors? How are they different from those of our closest living relatives? So to start off, I'll provide you with a little bit of what bones can tell us. They can tell us many things, and then I'll focus on what I'm particularly interested in, what my work focuses on. So we know that bones, we can infer an individual's age from the skeleton by looking at the dentition, looking at how the long bones have fused in the ends, We can infer sex, especially if we have the pelvic bones. We can infer or estimate stature of an individual or a population by looking at measurements of the long bones. We can tell or identify pathologies. We can identify diets through isotopic analysis and so on. Now, these last 2 points here that I have highlighted are that we can infer physical activity by looking at the human skeleton. And we can also, of course, look at the bones and see their evolutionary changes over a long time. And by saying that, starting with the physical activity, what I mean is that we can look at how dense a bone is, how the strength of that particular bone, and tell whether one was traveling long distances. Someone, walking every day for a long distance, making the thigh bone, for example, to be strong. Is an animal, for example, was it climbing trees as a way of means of locomotion or means of navigating its environment? So that is how, those are some of the things that we can infer in terms of activity. In terms of evolutionary change, we can look at, for instance, how curved or how structured certain bones are to identify relationships between species or between populations. And throughout the talk today, I'll be focusing on these 2 last points. What can bones tell us about physical activity, and what can it tell us about evolutionary change? Now, if you're wondering how we can infer activity, physical activity from bones in particular, I would like to just show you real quickly a nice illustration of how that change or how bones respond within a short time to activity. And prior to describing some of these images that I'm showing you down here, what we know by research, a whole century of research on bone morphology or how bone changes, we know that bone increases in density as we exert it, or if we put more stresses on it, increases in density. It also changes in its orientation in response to stress. Now, I would like to draw your attention to these images. So what we are looking at is a section through the hip bone of a fetus of a 3 to 6 month old, a 6 to 12 month old, and a 24 to 36 month old. And the white material that you see inside these images is bone. These are bony struts. And the black refers to, or it represents air spaces. And these air spaces are where in living tissue, in a living organism, we would be having marrow. Now, if we look at the fetus, you see a lot of blobs, a lot of white in there. There's no particular structure that we can identify. When we look at a 3 to 6 month old, there's more spacing. By 12 months, you see more spacing, and then you also see more bone in the lower portion and the upper part, if you can follow my cursor here as the pointer. And then by about 24 to 36 months, what we see is that bone structure is running in that upward downward trajectory, or what we refer to as superior-inferior pattern. Now, what's interesting about this is the age when children are starting to walk. And as they put stress on their limbs, or they put body weight on the hip bone, the bone is also orienting itself to resist those stresses. So this is just a beautiful, I love this picture because it's a beautiful illustration of how bone changes within a short duration of time. Now, you might be wondering then what are the mechanics that allow this to take place? Without going too much into detail, because a lot of these things, of course, physiological activity of cells is often quite complicated. I'm using this schematic or I'm showing you this schematic to briefly summarize what happens with bones. So basically, when we apply a load or a strain on our bones, such as when we are walking or when we are running, the cells, there are receptors in bone that will sense that strain, and consequently they'll activate cells within bone to start laying down more bone. as a way of resisting that stress. Alternatively, if we don't have any strain, or we are not using our limbs, for instance, at all, then another type of cell is activated to eat away that bone. So this is a continuous process that's taking place in our bodies, depending on how much loading or strain we put on our skeletons. So that's the brief summary of what happens, so the mechanics of how bone changes. Now, keeping in mind that interest I was telling you about in terms of changes in bone or in the skeleton that are short-term and changes that are long-term, I also wanted to show you, what's going on in the fossil record, because those, of course, those are, again, are changes that are occurring in the long term. And probably many of you are familiar with this, especially if you've gone to the National Museum of Natural History, the Smithsonian Museum, where they have a beautiful exhibit of some of our early ancestors. So what we are looking at here is the human lineage from about 3 million years here on the left, all the way to our own species here on the right. What's fascinating is that at about 2 million years, when we see the emergence of this species known as Homo erectus, this is when we see changes in anatomy that are similar to what we see in modern humans. So we see a change in stature, we see taller individuals, we see narrower bodies, so changes in body shape, changes in body mass, that some of which persist to our own species. So in my work, I try to link or to understand if some of these changes, one of them being, and I'll talk about this, is low bone density in modern humans, whether that is something that we also see in some of our early ancestors. Now, I've talked about the short-term changes, and I'm turning my attention to the long-term changes. Now, to summarize the changes that we see in the long term, and of course some of them are also short-term, we know that there are features that are uniquely human, and those features include things such as having a narrow pelvis, having long legs, having short toes, having large joints. All these is a suite of traits that we associate with our ability to walk upright. Now, more recently, some of my work and that of other colleagues seem to suggest that we, as in Homo sapiens, have lightly built skeletons. That means we have low bone density. And I'll be referring to lightly built or having a gracile skeleton as a way of expressing that low bone density that we see in modern humans. So the question is are these lightly built skeletons a result of low activity levels within our lifetimes, or is this an evolutionary adaptation, or even still an evolutionary mismatch? And I'll get to these here when we get to some of the questions that I've been asking, more detailed questions. Now, so far, I've been describing this, and maybe you're wondering why should we care about this? Why should we care about how bones change? For me personally, of course, I care about it because it's very exciting. It's very informative about our present, about our past. It's knowledge, and we're always seeking knowledge, isn't it? But in addition to that, there are some applications. And I'll tell you some of those applications that some people might have an interest. What we know is that some bone diseases, such as osteoporosis, is very prevalent, especially in the Western world. In fact, it's actually a public health concern, and that's because it is a disease that's characterized by low bone mass, and consequently, it increases fracture risk, which may lead to morbidity and, in some cases, even mortality. So, if we look at this graph here, what we are seeing is a plot of osteoporotic fractures, strokes, heart attacks, and breast cancer in the United States from about the years 2004, 2005, and 2006. And what we see is that osteoporotic fractures are quite high in number compared to the disease incidences. So this is just highlighting that public health concern that I was mentioning. But we of course know that clinicians and other bone biologists are really interested in questions of, what are the causes or metabolic causes of low bone mass? What are the dietary causes and all of this, as well as they're interested in identifying ways to mitigate these loss. But what I'm doing is, in a way, is trying to look at these concerns from an evolutionary perspective to see if what happened in the past is what is resulting in the trouble that we have now in this disease that is a major concern of ours. And again, I'll come back to that at the end when I've wrapped up some of these questions that I'll be talking about. So to address these, to address some of these things that I've raised regarding how bone changes with physical activity in the short term and whether it's related to changes in the long term, the main question I ask is whether the gracile skeletons we observe are a consequence of low activity levels, or whether it's an evolutionary adaptation or mismatch. And to get to that, first of all, we have to establish whether the pattern of gracile skeletons is indeed a recent evolutionary event. So right now, probably you're wondering, well, where do we begin to address this question? What are the methods we use? What parts of the skeleton will we be looking at? So I'll give you a brief representation of how we get to collecting our data to assess this. And often we use high resolution scanning, computed tomography scanning. So here, what I'm showing here on the left is a micro CT scanner. And this particular one is at the American Museum of Natural History. And basically we take our specimen, any particular bone of interest, we prepare it, put it into either in a container or in a piece of foam to stabilize it, and we put it into the machine. And the machine uses X-rays to gather 3D pictures and assembles it such that we have an image to analyze. The other machine that we use is known as the PQCT scanner, quite similar in terms of using X-ray imaging. And this picture actually was taken at the Smithsonian, at the Natural History Museum, perhaps 8 years ago. And now when we have these images from these scanners, what we do is we take a region of interest in the bone of it, in our image, and then we quantify the number of white pixels that represent bone, and then we divide that by the total number of pixels in a particular region, and that's what we end up with as our bone density. And that's what I'll be really referring to throughout this talk. Here are some additional pictures showing some of the bones that we've scanned, including this femur here of an early ancestor from East Africa. And it's, as you can see, it's inside, or maybe you may not be able to tell, but it's sitting inside a piece of foam. That piece of foam will stabilize it when the scanner starts rotating. And here is just an additional picture of me preparing some of these specimens in South Africa. And then down here in the bottom are pictures of some of the bones that we have that have been recently scanned, whose results I'll be showing you today. So this is from the one I'm pointing at on the far left is the skeletons from the Czech Republic. These are Homo sapiens from the Upper Paleolithic period. That's about 26,000 years. And then here we have some from Israel. These are also Homo sapiens from about 100,000 years ago. And then we have the resulting, just to show you what a fossilized bone image would look like. So this would be the inside. This is the inside of one of these really fossilized bones. And what you see is a lot of gray and white. And this is the trouble of working with fossils, is that you end up with an image that's not very distinctive in in terms of what is bone and what's not bone. So it requires a lot of work in the lab to clean up these images so that we can tell what's bone, which is the darker material, versus the air matrix or air spaces, which in this case are filled up with fossilized or minerals and soil. So that's just a quick glimpse of what we deal with in terms of scanning, in terms of specimens. Now, to get a full representation of the skeleton, so if we are going to really ask these questions, we have to sample all the bones that we can get, or at least most of the bones that we can get. So in the upper limb, we've sampled the upper arm, the arm bones, the forearm, bones of the hand itself. In the lower limb, we've sampled the hip bone, which is the femur, the tibia, which is the leg bone, and some toe bones. So that's the representation of the data I'm going to show you today. But sometimes we've added, or in the past I've added vertebrae, that in the fossil record, those are usually a bit harder to come about, and the right bones to scan can be a little bit more challenging to identify. So to answer that first question about changes within our lifetimes, depending on our physical activity, what we did is we sampled modern humans with different subsistence strategies. And when I say modern humans, I'm referring to people that lived in the last 10,000 years. So these are samples from the archaeological record. And some of these samples are actually housed at the Smithsonian, but also includes specimens from other museums too. So we had farmers, we had hunter-gatherers, included industrialized people or populations, and then fisher foragers. The presumption here is that if you are a farmer or someone that worked in, has lived in an urban industrialized environment, you are relatively sedentary because you're either going to the farm and working in that farm and returning home, or you are going to work in a factory, for instance, and returning home. If you're a hunter-gatherer or a fisher forager, you are, of course, traveling long distances to gather your food, or to hunt for game, or you're on boats, you're rowing to go and capture you kill your fish, the big mammals, and so on and so forth. So there's more activity taking place. So I refer to these two, the hunter-gatherers and fisher foragers, as active, while the farming and industrialized groups are more sedentary. So I'll show you a few results here of these comparisons. So if we look at compare the hunter-gatherers and foragers versus farmers and in the industrialized populations, we see that, let me orient you here on the graph, on the box plot, we see that the foragers actually have greater bone density than the farmers and the industrialized groups. And these box plots, the bold line in the center represents the median. And the long tails that we are seeing is the range of variation. So when we pull these samples of the sedentary versus the foragers, we actually see quite a significant difference. So this really affirms or supports our hypothesis or idea that if changes are taking place within our lifetimes, and if you're more active, the more bone density you have. And this is not just my work alone, but also work of other colleagues that illustrated this very nicely in this image. So if we look at these hip bone of a hunter-gatherer compared to an agriculturalist, we see the white material, which is bone, it's quite dense in here. There's a lot of it. And the actual thickness is big. When we look at the agriculturalists here, we see that there's more sparse and thinner bone. So again, affirming that idea of change within our lifetimes. But then the other question is, how does this translate to long-term, large time scales, or long time scales, if you will? So to get to that, the long time scales or evolutionary change, what we did is we took our humans, the ones I just showed you in the previous slide. So these are the recent modern humans, sedentary ones, and then we have our active recent humans, and we compared them to those upper Paleolithic humans from about 26,000 years ago from Europe. Then we compared them again with early modern humans from Israel from about 100,000 years ago, compared them to Neanderthals from Europe from roughly 60,000 years ago. And then we have our sole early Homo, an individual from about 2 million years ago. So I should just mention that early Homo is our genus, but not our species. We think this is a Homo erectus. Neanderthals, of course, are not our species, but same genus and closely related. And then we have the rest are all our species, but earlier, occurring earlier in time. And the pattern that you note is that these two modern humans ourselves are quite low in density compared to the rest, regardless of species. So this is really, really interesting because it confirms or affirms the idea that these gracile skeletons are a recent evolutionary event. And at the same time, there's a correlation to activity. If you're more active, you're foraging, you have more density. If you are farming or you are industrial, you have low bone density. So this is great because it's just the beginning of a lot of more hypotheses to be tested, and as I'll show you in the next few slides. So in addition to that, perhaps you already saw this image from the announcement that went out for this talk. This is an illustration of some of the comparisons that we made, including chimpanzees, our closely living relatives, and some of the older fossils from South Africa in particular. And when we compare them, the chimpanzee, our human ancestor, Australopithecus africanus, some of the early modern humans and recent humans, we see a pattern in which, again, like I mentioned before, that recent modern humans are unique. And I'd like to show this image because it illustrates this fact very nicely. In particular, if you look at the bottom pictures, we see that, so these are sections through the hand bones, and the hand bone of a human has very sparse bone. It's quite porous compared to that of a member of our own species that occurred a little bit earlier, which has more bone, and of course, very distinctive from this early ancestor, and as well as the chimpanzee. So again, reiterating that recent change in bone density. And the thing that, of course, we note, or the most important thing that coincides with this low bone density, is the fact that in the last, when we are seeing this change, this is a time period when we have a significant shift in behavior of modern humans. This is the time then when we start domesticating crops. This is the time we start domesticating plants. This is a time when we start living in settlements, and of course, eventually living in city-states and becoming, the beginnings of these sedentary lifestyles that we lead now. So returning to those questions that I started out with, are gracile skeletons a consequence of low activity levels or evolutionary adaptation? At the moment, I would say it's likely both, because we see change depending on what you do in your lifetime, and also we see change in the last 10,000 years. Right? So there must be something that's causing this shift. That's not just change that we do within our lifetimes and that's the end, right? It's change that is actually affecting our genetic patterns, or it's being passed on into populations. So a better word is that it's a systemic change. Then the second question, or the sub-question, if you will, is whether the pattern of lightly built skeletons is a recent evolutionary event. All the data so far that we've gathered, that others have gathered, indicates that it is indeed a recent evolutionary event. Now, returning to applications of some of this work, I think looking at skeletal changes from an evolutionary perspective does not conflict or does not divert away from physiological research that's going on bone diseases, but it really does complement that work, and it helps us understand a little bit why we are vulnerable to bone disease. Because in evolutionary terms, of course, there are mismatches. There are features that are advantageous and there are features that are disadvantageous, and there are features that are both advantageous and disadvantageous. And as I tell my students in our evolution course, that these are things that they are trade-offs that we see in our skeletons or in our morphology throughout human prehistory. Now, some of this work has been highlighted a little bit in the last 5 years or so, showing that we have light skeletons due to less activity, that the agricultural innovations are what led to these light skeletons. And I just want to add that, well, to say, yes, I think this is the case, but at the same time, it opens up windows to test other hypotheses. This systemic change, I believe, is probably more than just an agricultural, we become sedentary and then we have light bones. I do believe there's more to it, which of course opens up more lines of investigations that I continue to do. So in summary, through my work and work that I continue to do, we need to thoroughly document the changes, or to answer these questions, especially relating to being sedentary and how that affects our bones. While we have data that has shown that this is not the end of it, we still need to thoroughly document these anatomical changes consistently in the last 10,000 years from different regions, different geographical regions, so that we can have a better insight into this shift. And one of the other exciting things that I'm working on, that I think the work that I've just shown you opens up, is to try and use other mammalian analogs to document changes similar to those seen in modern human dogs. And in particular, I'm interested in dogs because I think they present an analog that would inform modern human anatomy. So I'll show you just a little bit of the preliminary work that I've been I'm working on right now, which is comparing domestic dogs to wild canids. So if you're wondering why domestic dogs and wild canids, is that we know that these are species, or these are organisms that were domesticated in the last at least 15,000 years. Well, I guess we can have debates about when exactly that happened, and people that study dogs obviously have a lot to say about that. But generally speaking, the last 15,000 years, dogs were domesticated. We know their closest real living relative is the grey wolf. So how do those two compare? Does domestication have an effect on bone density? So what we've done is compared the grey wolf to a variety of breeds of dogs, including the husky, the greyhound, the Saint Bernard, and the Eskimo dog. And this is the last graph I'm going to show, I promise you, showing bone density in the wolf and the domestic dog. And what we see is that overall, when you look at the means as well as the median, statistically speaking, we see that the wolf has more bone density compared to the dog. So you might say, well, this is the median, and you have a large range here. But even with a large range, the domestic dog has lower bone density than the wolf. So this is really interesting. So it really doesn't matter. Of those 4 breeds we compared, all of them have low bone density than the wolf. And keep in mind, we have the husky there, an animal that's bred for, the dog bred for endurance. Keep in mind, we have the Eskimo dog there, also a dog that's been used in endurance. And these are, especially the Eskimo dog was obtained, or was a collection, is from the American Museum of Natural natural history, a collection that was acquired from the 1900s or thereabouts. So this is really, really fascinating, and I'm really excited about this. And I think it might be able to tell us something about our own so-called domestication, if you like, because there's been this lovely proposition or hypothesis that's been put out that modern humans underwent our own self-domestication in our own way. And if that's, for example, settling and being cooperative and living in societies was an advantageous trait or an advantageous behavior, perhaps some of the byproduct of that behavior is having a gracile skeleton, as has been shown in some other species of dogs, of carnivores, if you will. So that's just, if people have questions about these, I'll be happy to talk a bit about it, talk a little bit more about it afterwards. But I just wanted to touch on it a little because it's quite, I think it's quite interesting. So to summarize, so far I've shown you that gracile skeletons seem to be a recent phenomenon through comparisons of human ancestors and recent modern humans, which are showing that we have light skeletons, and these light skeletons seem to have occurred quite recently. And these results, or these conclusions, have implications for understanding evolutionary adaptations. Are they related to domestication? Are they related to our sedentary behaviors? Are they? How does it fit into the origin, the bone disease? Was it an advantage that led us to be more vulnerable to bone disease in such that, and I'll give you an example, such that if you have a lighter skeleton, for instance, there's an advantage there in that you can move it more easily. You have less mass to move around, right? But on the other hand, perhaps you're pushing the extremes evolutionarily to the point that you're more vulnerable to bone disease, or there's increased porosity, so to speak. Then lastly, that sedentism plays a role in the emergence of light skeletons. It seems that it does play a role, but there might be alternative explanations, such as what I was describing as the self-domestication hypothesis. So with that, I would like to conclude and end by thanking all of you for being in attendance today. And I would like to thank the Human Origins Program, particularly Dr. Brianna Pobina, for inviting me to participate today. Other folks at the Human Origins Program, Dr. Rick Potts, Jenny Clark, people that I've worked with in the past. Thank you very much for all you do and all your support, and many other folks, including Naima, for organizing this, and curators from various museums and institutions that have allowed me access to specimens. So thank you.
Wonderful. Thank you, Habiba. That was fantastic. And thank you for giving us a tantalizing glimpse into what you're working on now regarding the self-domestication hypothesis. So we have several questions. We will get to as many as we can between now and 12:30 when the program ends. The first one that I'll ask you is a pretty basic methods question, and this is from Mercedes. Is it necessary to handle the bones using gloves?
So for modern, so it depends, I would say. It depends on what specimens you're dealing with. So modern human skeletons often at museums tend to be relatively well preserved, so you don't need to handle them with gloves. The fossils, that's another, it also depends on what kind, where they were found and their state of preservation. The ones I've had to handle so far, it's not necessary because they're highly fossilized. So it's almost, they're almost rock, like rock. So it's not necessary to use gloves. But I know in other areas where fossils have been found in caves, the bones might be crumbly and they might be very delicate. So it might be necessary to use gloves.
Okay, thank you. Here's a question from Heather. Bone density or structure changes in childhood. Does it continue to change in adults?
Yes, it does. It does, depending on many factors. Some of them I've not talked about, of course, dietary, hormonal genetic factors. But the one that I have touched on is activity, and it changes with how much activity you put on your skeleton. So for instance, if you are an active child into your teenage years, you're an athlete, for instance, your bones will be stronger, greater bone density, and that density will actually persist into your adulthood, even if you stop being very active. So you see low prevalence of, say, osteopenia and osteoporosis in people that were really active during their teenage years when they're growing. So yes, our bones do change throughout our lifetimes.
Excellent, thank you. And I'm going to ask a follow-up question because somebody asked about an athlete. So this is a question from CJ Green. In the slide of the gracile skeletons being a more recent evolutionary event correlating with activity, where would an Olympic athlete fit?
That's a really good question. And I haven't done the studies of athletes, but colleagues have done it, not looking at particularly at bone density, but at bone strength. And what they showed, actually, it's a paper that was published in, I think, in the Journal of Human Evolution. They showed that athletes were similar to these closer to these the upper Paleolithic humans that I showed. So yes, they do increase, but again, keep in mind our upper Paleolithic humans, so the samples that we have are quite small. So we might look at 2 upper Paleolithic humans and compare them with 5 athletes and see they're quite similar, but they're not, we don't know, I wouldn't say we have a full picture. But yeah, they tend to be quite strong. And on the other hand, also it depends on which bones you're looking at. So I think in that study, what they looked at was I think they looked at people that were rowing. Yeah, people that are using their upper limbs more, and it seemed to show in their upper limbs that they were quite strong. So.
This is a nice follow-on question. This one and the next one I think I'll ask are sort of about predictions. So Stephen Rockauer asks, is the relative osteoporosis you see now reversible? If we were to live the sort of life of early humans or Neanderthals, would our bone density respond appropriately?
That's a good question, but a hard question to answer. And that's because, of course, we know our bones will change throughout our lifetimes, depending on how much activity we exert. So yes, in that case, we would. But in terms of a change, a long-term change, to be like our ancestors, I do not know. That's a little bit the reversal of the evolutionary process. I couldn't speak on it. But just change within our individual people, within their lifetimes, yes, we can exercise a lot and build up our bone density. But in terms of population-wise, I couldn't say too much.
Okay, that's fair. Here's a really interesting question from Sophie QB. If a child grew up in a spaceship without gravity, would their bones not show that pattern at 36 months, but look more like a younger one?
Probably, I would say yes, because you're not, without gravity. So the pattern of growth your cells are not, and the mechanoreceptors are not sensing the weight, the stress. So most likely problematic, diseased type bone or skeleton, if you will.
Okay.
Yeah, that's really interesting, actually.
Yeah.
Think about it. With all these discussions of space, of
exploration. That's true. Yeah. Colonization, maybe.
Colonization, if you will. Yeah.
So here's a question from Suzanne Ubik, who asks, in industrialized societies, have comparisons been done between manual workers and heavy industry workers and people who don't rely on physical labor for their income?
Not that I know of. And the problem is, I think it would be really interesting to make these comparisons. The problem is actually using or collecting this data in living people. You see, so you can use equipment that clinicians use, for example, a DEXA scan to do that. But the people that often, clinicians are more interested in diagnosis and of individuals, or maybe just a fewer samples. But people that are more interested in broad change or bigger patterns, I do not within at least not in the close in biological anthropology or paleoanthropology, not that I know of. Sorry,
Okay.
can't answer.
No, that's okay. So here are two questions that are kind of similar. One's from Christina. Do you think changes in dietary intake when we relied on agricultural products in place of forage foods might lead to reduced calcium or vitamin D intake, thereby contributing to reduced bone density? And
Yeah,
CJ,
sorry.
and similar question, CJ Green asks, what do you think the impact of eating or drinking dairy has had on this? Isn't it a modern human thing? Could it actually leach calcium from your bones? And what about the impact of fluoride in our diets?
So to answer that, both of those, well, I'll start with the first one, which was a bit more general. Yes, it has an effect. Diets have an effect on bone density, as well as, of course, teeth. And there's some work showing that as soon as we started using corn, we are domesticating all these grains, you see a change in, there are more pathologies, our dentition pathologies, our skeletons. So these really could be a contributing factor. But I would say this, the pattern that we are seeing seems to be consistent even among populations that were not engaging in agriculture, or not farming, right? We have the foragers. The foragers have strong bones, but they still are not as high density as our ancestors. So there's something there. I think diet plays a critical role, but I think there still are something else beyond just diet. In terms of dairy, yes, together with including more grains in our diets, I think it would have an impact. How much of a leaching they would have on calcium, I couldn't say, but overall, the process, the change in diet has been documented to be using more dairy and using more grains in our diet has been shown to increase pathologies in particular, to lead to increase in pathologies.
Thank you. Here's a good question from Hannah Jacobson. If you know this, how quickly can bone density be remodeled? For example, would our skeletons be different over a 6-month period from the beginning of quarantine, where maybe we had an active lifestyle, to now a less active lifestyle?
Well, so if you think about it, actually it does happen quite fast. So if you think about having if you break your arm or break your leg, any of your bones, right? It will heal, that bone will heal over, say, well, we don't want that, but say you did not do anything about it, right? You did not see a doctor. That bone will heal over within a duration of a number of weeks, maybe 4-6 weeks, it will have grown over where the break happened. So yes, it does remodel fast. But I couldn't speculate on what quarantine has done for us, or to us, I should say. But I would, the example of the bone break is also an extreme situation in which the body physiologically is trying to heal itself. So there's a faster mechanism taking place compared to, say, just, walking from the living room to the kitchen and, you're still, there's nothing wrong with your body. So there's no compensation to heal it or to remodel it. But I would say it does occur, not in a long time. And that in that picture I showed you of the kid, well, the child at about 2 years, they're starting to show, you see that change in orientation, in the distribution of bone, in the orientation of bone. So yes, within a short weeks to, years, obviously, in that case.
Excellent, thank you. So here's a question from Kimberly Tommy. Did you look at a specific breed of domesticated dogs? And if you did, why that breed?
Yes, I did. I did look at specific breeds. So I had greyhounds, I had Saint Bernards, I had Eskimo dogs, and then we had Huskies. Now, the reason, well, there's a pattern, or there's there's a reason for this, and there's also, how do you say, availability of specimens. So the huskies in particular, I was really interested in them and the Eskimo dogs because of their endurance. Because part of what I was trying to get at is whether you can look at differences between, say, dogs that are bred for endurance versus dogs that are bred for grass, that are quite gracile, like the greyhound. So that was part of the reason I was getting at. But at the same time, it was also availability of the collections of the specimens at the museums. Because when you go to museums, it's hard to find known breeds of dogs. You just find, well, Canis familiaris, that is dogs all together. But the specific breeds are usually hard to find. So I was lucky enough that the Smithsonian has the greyhounds, and a few other breeds. And then the American Museum of Natural History had the Eskimo dogs and St. Bernards, and the Field Museum in Chicago had the Huskies. So yes, so yeah, so it's both, but availability as well as there were reasons that I was trying to get at. But or rather, in addition to, what we are trying to do is also add in the dingoes. So I think that would make it really interesting, because if you think about dingoes, probably they're a bit more wild, so to speak, and we have dingoes from the archaeological record from about 5,000 years ago, if you can believe it. So that's pretty neat. So yeah, I hope I answered that question at least partially.
Yeah, thank you. So Cecilia Thonet asks, in addition to activity, what other factors account for the bone changes or structures from primates to modern humans? She asks, diet, heredity, age?
All the above, all the above. Yeah, so activity, of course, well, let me just go back and start with activity and then get to the others. For other primates compared to humans, of course, say, for example, monkeys, right? They're arboreal, they're climbing trees, so you will see patterns that are completely different from ours. In terms of diets, it's harder to say between primates and humans, but then again, like I was mentioning, in modern humans, in the archaeological record, you see that there's an influence of diet, or rather certain diets resulting in some known pathologies. In terms of heredity, yes, you see that if you look at all primates and have compared chimpanzees, orangutans, baboons, some monkeys, different types of monkeys, you see that generally, while there are variations between species, they all tend to have high bone density. So of course, there's a heredity or phylogenetic signal there that might be going on. Did she mention a third one?
I think it was age and heredity and something else, maybe diet.
Yes, and age too. Yes, I did touch on age. Yes, age is important, and that's why we try as much as we can when we are sampling modern humans to sample at least individuals of 40, if the age is known, of 40 and below, or 45 and below, because after that you really just don't know what you're getting at if the individuals are a bit, had osteopenia or the beginnings of osteoporosis. So yeah.
Thank you. So I think you actually just answered the next question I was going to ask. This is from Sophie QB. Are all the skeletons that you have scanned to compare the same age? If some are older than others, could that affect the comparison? And I think she means sort of biological age as opposed to geological age.
Right. Yes, absolutely. We try as much as we can. There are some, of course, from the archaeological record in which you couldn't tell the age. So that presents a challenge. But what we do, or what I've done in the past, is if I see that there's a bone that presents itself as obviously just looks a bit more pathological, or when you hold it's a bit lighter, I exclude it from the analysis or from the scanning. So as much as possible to try and scan anything that's 45 and below. And the Smithsonian has some wonderful collections there that of known age. So that always helps when you have known age, and other museums too. But when it comes to the archaeological record, it's a little bit more trickier, but we try.
Great. Here's a question from Lisa Berberian, and I apologize if I mispronounced your last name. Dr. Chirchir, have you done anthropological research on the evolution of human bones in Kenya? And if so, did you discover anything distinctive about the fossils in Kenya compared to elsewhere?
Yes, so it's really preliminary. I'm very excited about this. So last year, we were able to scan a couple of bones of human ancestors, of fossils, from northern Kenya. And these are members of Homo erectus and what's called Paranthropus, okay? And I was hoping, my expectation was that, well, Homo erectus will be more similar to us in having low bone density, but no, it has high bone density. And if you like, I don't know, Brianna, if we have time, I can show that graph a little bit one more time. Do we have time?
Sure. And after this, we'll have time for probably one or two more questions.
All right, let me just share that screen, and I hope it will
Sure.
help the person that asked the question. So if we look at this graph, this early Homo, this is a Homo erectus. We think it's a Homo erectus, but either way, it's identified as a Homo species. And you see it has high bone density. It's a single specimen. Compare that to modern humans. So it was really, really interesting. Because I thought it should be maybe a little bit closer to modern humans. Now, the other one that I just talked about, I'll stop sharing the screen now. The other one that I just talked about, the Paranthropus also had bone density, and that was high bone density, and that wasn't too surprising. But what we plan to do, have I stopped sharing the screen, Brianna? Sorry.
Now you have, yeah.
Okay. We were able to only scan those two because we only had permission to scan the two. It's a long process. We had to get permission to get the fossils and travel to South Africa to scan them and then return the fossils and then leave with the data. But now we are starting a new project to scan more of those fossils from northern Kenya. So this is really going to be something I'm really looking forward to getting those results. So to answer your question, yes, we have, but only two, and both of them show really high bone density.
Ooh, interesting. Well, that's exciting research in progress. Okay, so I will have one last question, which is basically sort of questions from two people combined. So this is another prediction question. Shannon Hathaway says, I teach 6th grade social studies. We start the year learning about early humans. My students always ask what we would look like in the future based off of how much we've evolved over time. I would love to hear your thoughts so that I can talk with them. Thank you. And Christopher asked, what will our bones look like in the near future?
So when we think, I don't know if I have a straightforward answer for your students, because it's a bit of I guess a harder question to, especially if you're working with kids in which you want to break down some of these responses. But if you think about the evolutionary process, right, it's a continuous process, and we really don't have a direction. We don't know what is advantageous or disadvantageous, what's a byproduct of a particular selection. So it's really hard to say. But what I can say, generally speaking, is that we are changing, and most likely in years to come, after we are out of this world, there'll be creatures that probably are somewhat similar to us, maybe a bit different from us, because this process is continuing. So I couldn't speculate on how we will look like, but of course the things we do may have an influence on whatever the next species that comes about, or just the same species evolving into something else. So hard to say.
It is, no, and I think those prediction questions, especially about the future, are really difficult. Okay, I will ask one last question from Platonov. Can you explain a bit more about the self-domestication hypothesis?
Okay, yes, this is really interesting, and some very clever researchers came about to this. And basically what it postulates is that as people, as human beings, became more cooperative and started working together, cognitively cooperative, working together in communities or groups, in social groups, the need for these large bodies and more strength is diminished. So more cooperation leads to, one of the there was more cooperation, and one of the byproducts of that selection for cooperation was changes in our physiology and in our anatomy that was not expected. It wasn't part of the selection process. And I'll give you an example of something that's part of where this came from. Is that there was this famous study that was done in Russia of foxes. And basically what was done is that these researchers bred tame foxes in one line and aggressive foxes in one line. And what they were selecting for a particular behavior, right? Tameness or aggression. And what they noticed, which they were not thinking about, was that there were changes in the physiology as well as in the morphology of these foxes. The aggressive foxes just tended to have, in terms of morphology, had slightly larger teeth, had slightly larger jaw lengths and skulls, while the tame ones tended to be more like dogs. They were very puppy-like, had curly tails, had floppy ears, droopy ears, if you will, and so on. So the speculation or the hypothesis is suggesting that if the friendliness and cooperation affects neural crest cells, cells that occur during development, that also have an impact on our morphology. So it's a really, really interesting idea. And perhaps that's what's happening to us, because what we see, and if we have time, I have a slide I can share about this. If we have a minute, and if the person that asked the question is still on here. Can you see that?
Yeah.
Okay, so what you see is that even in modern humans, in the last 10,000 years, you see a change from these robust bodies to gracile bodies in the face, in the thickness of the cranium, of the cranial bones. So this is the origin of these humans, or the ideas behind these human self-domestication hypotheses.
Cool, thank you. So we have run out of time. And so first of all, thank you for the wonderful presentation. I apologize to everybody whose questions I wasn't able to get to. Thank you to everyone in the audience for asking wonderful questions. As I mentioned in the beginning, so my name is Brianna Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. This is a part of our monthly HOT topic, which stands for Human Origins Today program series. Those of you who registered via email, you will be getting emails about our future HOT topic programs, which we generally try to schedule on the third Wednesday of the month at 11:30 a.m. So stay tuned for those. And I just want to thank Dr. Chirchir again for a really fascinating presentation, for being able to answer all these rapid fire questions. And thanks to everyone in the audience. All right, take care. Bye, everyone.
Thank you, everybody. Thank you, Brianna.
Thank you.